Welcome back to the Science Channel podcast, where we discuss the coolest scientific papers and preprints of our day. If you're finding these explorations into cutting-edge science interesting and helpful for your understanding, please do give us a like and subscribe. It really helps the channel grow and allows us to bring you more of this content. Today we're venturing into the fascinating world of cellular aging, looking specifically at a study you shared with us that delves into why even seemingly identical cells can have dramatically different lifespans.
Oh yeah, this one's really cool.
Yeah, we're focusing on the single-celled yeast, Saccharomyces cerevisiae, a workhorse in biological research.
A classic model organism.
Right. So when you look at a population of these yeast cells, all with the same genetic makeup and living under identical conditions, you'd expect them to age at a similar pace.
Right, you'd think so.
But that's not what we observe. There's a surprising amount of variation in how long they live.
Yeah, there really is.
And this particular study by Hilsebeck and Rhea that you brought to our attention tackles this puzzle head on. Their core idea is that the intricate network of metabolic reactions happening inside these tiny cells, the different routes and rates at which molecules are transformed, what they call metabolic flux distributions, could be the key to understanding these lifespan differences.
That's the idea.
Okay, so if we think about the inside of a yeast cell, like a super busy factory with thousands of different production lines and delivery routes,
Okay.
these researchers are suggesting that the specific way this factory operates, the flow of materials and the speed of each process, might determine how long the factory can keep running.
I like that analogy.
To investigate this, they didn't just take a quick look, they really immersed themselves in the details. The study utilized a massive computational model of yeast metabolism, Yeast GMM version 8.5.0. This model is incredibly comprehensive, including information on 1,150 genes, over 4,000 different biochemical reactions, and more than 2,700 metabolites.
Wow.
It's essentially a complete virtual blueprint of the yeast cell's metabolic engine.
Yeah, and these models, they're like the unsung heroes of systems biology, right?
Yeah.
I mean, they allow us to simulate and predict how a cell might behave under different conditions, which is just incredible.
What's fascinating here is how they use this detailed blueprint to explore the link with lifespan.
Exactly.
The researchers systematically went gene by gene, effectively turning off each of those 11,150 genes one at a time for each gene knockout that resulted in a viable yeast cell. And there were 812 of them. They then used the model to generate a vast landscape of possible metabolic flux distributions.
Okay.
Over 406,000 different ways the cell could potentially operate metabolically.
Wow, so for each of those 812 viable mutants, they simulated hundreds of different ways the metabolism could be rewired?
Exactly. It's like having thousands of different operational manuals for each slightly modified factory.
That's a great way to put it.
That's an astounding amount of data.
It is.
So for each of these 812 genetic tweaks and the resulting range of potential metabolic activities, they then went into the lab to collect real-world lifespan data. They meticulously measured the replicative lifespan of individual yeast cells. That's the number of times a mother cell can divide and produce daughter cells before it stops.
Okay.
In total, they tracked the aging of over 66,000 individual yeast cells.
So they had this huge experiment running in parallel, a computational one predicting metabolic possibilities, and a biological one measuring actual lifespans.
Exactly.
Really impressive.
And the challenge then becomes, how do you connect these two massive data sets? How do you find the signal in all that noise?
Right, because you've got all this data, you need a way to make sense of it.
And this is where their sophisticated analytical methods come in. They employed principal component analysis, or PCA, which is a way to take a very complex data set with many different variables and reduce it down to a smaller number of key underlying patterns.
So sort of like finding the principal axes of variation in the data.
Right. Think of it as finding the most important levers that control the system.
I like that analogy.
They also utilized various deep learning approaches, different types of artificial neural networks, including regression neural networks, classification neural networks, and even convolutional neural networks.
Now, CNNs, those are usually used for image recognition, right?
Exactly. So how were they applied to something as abstract as metabolic flux?
Well, they did something quite clever. They developed an algorithm to transform those complex metabolic flux distributions into a kind of pixelated image.
Okay.
Imagine each pixel representing the activity level of a particular metabolic reaction.
I see.
This allowed them to leverage the pattern recognition capabilities of CNNs to see if there were visual signatures in the metabolic activity that correlated with how long the yeast cells lived.
So they weren't looking at pictures of yeast cells, but rather visual representations of their internal metabolic states.
Precisely.
That's a fantastic way to visualize it. A metabolic fingerprint.
I know, it's really cool.
So with all these powerful analytical tools pointed at this incredible amount of data you shared with us, what was the key insight? What did they discover about why these yeast cells age differently?
Well, the central and quite striking finding is that across all 4 of their different analytical methods, the researchers consistently identified a core network of interconnected metabolic reactions that appear to be the primary drivers of the aging rate in yeast.
Really? And this core network, based on their analysis, seems to be sufficient to account for all the variation in lifespan they observed in their experiment.
All of it.
All of it.
That's a really significant claim.
It is. So when we talk about this core network, what kind of metabolic processes are we focusing on? What are the key players?
The study highlights several important biosynthetic pathways.
Okay.
These are the cellular assembly lines responsible for creating essential molecules like ceramides, monolysocardiolipins, phosphoinositides, porphyrin, and glycerolipids. These molecules play vital roles in everything from cell membrane structure and signaling to energy storage. The fact that these specific pathways kept emerging from their analysis strongly suggests that the way a cell manages the production and processing of these particular lipids is crucial for determining its lifespan.
So it's not just about how quickly the cell is growing overall or how much energy it's producing in total. It's about the specific pathways and the precise rates of these interconnected metabolic reactions, particularly those involved in lipid metabolism, that really dictate the pace of aging at a cellular level.
That's the key takeaway.
So their findings led them to propose that the variation we see in the replicative lifespan of yeast isn't just random biological noise. It's an inherent property that arises directly from the very structure and operation of the cell's metabolic network.
That makes sense.
Furthermore, their data indicates that despite the vast number of possible metabolic configurations, these configurations tend to coalesce around 3 main metastable states.
Metastable states.
Yeah.
That's an interesting concept.
Think of it like a ball rolling across a landscape with several valleys. The ball will tend to settle into the bottom of one of these valleys, representing a relatively stable state. In this case, the researchers identified 3 such metabolic valleys, one associated with an extended lifespan for the yeast cell, another linked to a shortened lifespan, and a third representing a more typical wild-type lifespan.
So it's as if the cell's metabolic machinery tends to gravitate towards one of these 3 general operational modes.
Right.
And that choice then heavily influences how long it will survive.
That gives us a really clear picture of these almost preferred settings for the cell's metabolic engine that have profound effects on longevity.
Yeah, it's pretty mind-blowing.
It is. So for you following along, the key insights from this deep dive into the study you shared are: the differences in lifespan among even genetically identical yeast cells appear to be driven by the inherent variability in their metabolic networks. These metabolic states seem to settle into 3 main, relatively stable configurations, each linked to a different lost fan outcome. And the study specifically identified lipid-related pathways, like those producing ceramides and glycerolipids, as being particularly critical in controlling the rate of aging. Plus, the fact that multiple independent computational approaches all pointed to the same underlying metabolic controls really strengthens the confidence in these findings.
Absolutely. And it's worth noting a somewhat surprising finding.
Okay?
The researchers pointed out that the reaction in their yeast model that represents the overall production of biomass, essentially how fast the yeast is growing, was not identified as a key controller of lifespan. This really emphasizes the point that it's not just about the general metabolic rate, but the very specific routes and decisions the cell makes within its metabolic network that are most important for determining longevity.
That's definitely counterintuitive.
It is.
You might initially assume that slower growth would always correlate with a longer lifespan. But this suggests a much more nuanced picture, where specific metabolic choices are paramount.
Yeah, it's not just about slowing things down, it's about making the right metabolic choices.
Now, as with any complex study like this one you brought to our attention, there are always limitations and caveats to consider right.
Of course.
The researchers themselves acknowledge that they used a specific version of the yeast metabolic model, version 8.5.00, which, while incredibly detailed, is still being refined. For instance, they mentioned that the modeling of lipid metabolism in this version has some known limitations.
Right, lipid metabolism is notoriously complex.
And as our understanding of yeast metabolism continues to grow, and newer versions of the model, like Yeast 9.0, incorporate more data and improve accuracy, we might see even more precise insights into this core aging network.
Definitely.
So this study provides a valuable snapshot based on the current state of our knowledge. And future advancements in our understanding of yeast metabolism could further refine these findings.
Absolutely.
What about where this research might lead us in the future? What are some of the exciting potential directions?
Well, this study has really opened up some compelling avenues for future exploration. The methodologies they developed, combining comprehensive metabolic models with sophisticated machine learning techniques to predict lifespan, could potentially be applied to other organisms for which we have similarly detailed metabolic models.
Oh, that would be interesting.
Wouldn't it? It would be fascinating to see if this concept of tri-stable or perhaps even more complex lifespan states holds true in other species as well, maybe even in human cells. Further research could also focus on understanding the precise mechanisms that drive these metabolic configurations towards these specific stable states.
It really makes you think, doesn't it? Could the inherent randomness in the metabolic choices within our own incredibly complex cells be a fundamental reason why we all age in different rates?
That's a deep question.
It is. And if that's the case, could we one day find ways to influence these metabolic pathways, not just to extend our lifespan, but to promote a longer and healthier life?
That's the ultimate goal, isn't it?
It is.
To not just live longer, but to live better.
It's certainly a thought-provoking conclusion to draw from this deep dive.
Indeed, it suggests that the fundamental processes of aging might be deeply intertwined with the intricate and dynamic world of our own internal metabolic operations.
Well, this has been another fascinating deep dive, exploring the metabolic underpinnings of aging in yeast based on the study you shared with us. Thank you for bringing this intriguing research to our attention.
It was my pleasure. It's always rewarding to delve into these complex studies and uncover fundamental biological principles that might have broader implications.
Please don't forget to like the video and subscribe if you want more content like this.